generate some waste, just a few smart cells, not much more than that. The actual amount of reagents is almost nothing compared to what our competition uses. This blade center is where we have, a pretty strong computer system. In the blade center, I'll talk about it in a little bit, but all of the data has to go through the blade center. The cameras and stuff streams all of its data into the blade center. Another movie here, this is just a quick short one. This is looking into the top half of the instrument, and this shows you how the robotic kind of runs around on the work deck, which is what we call the top half. So you've got pipetters in the robot, which will take the DNA. You've got grippers in the robot, which will transfer the smart cells around the work deck when maybe you want to put your magnetic beads in or whatever. Ultimately, they take it over to this, which is what we call the stage. And this has got 3 legs on it. And the stage is where you drop the smart cell off, and then you have to put a capper over the top, which will seal it environmentally. And that's where the actual sequencing occurs.
So sorry, is there only one smart cell in the machine? Or?
Only one. We can only process one smart cell at a time with this instrument. Some of the aspects of some of the subsystems, I'll talk about this, some of these just a little bit. The stage itself has 6 degrees of rotation, and really what the stage is designed to do is to take the smart cell, anchor it solid, and then hinge it, align it perfectly to all of the beamlets. And I'll talk about that in a little bit. The grippers move around. That's all part of the robotic aspects. We've got pneumatic shocks. I'll talk about the optics, but the optical subsystem, we've got over 30 feet of free space optics designed into this thing. And so we can't do very well with shocks during the system. I already mentioned some of the fluidics that we've got. Not a whole lot in volume, but we do have to handle the fluidics that head around. The environmental systems kind of tell you a little bit more about some of the challenges. So the optics, if we move more than 0.1 degrees C per hour, it pretty much hoses our optics and our alignments. Oxygen will kill the polymerase, so you have to make sure that if there's any O2 in the suspension with the polymerase or on the smart cell, that we have to get rid of that. In fact, we drop enzymes. There's oxygen scavenging enzymes that we actually put into the suspension that will kill off any O2. And then on top of that, we use humidified nitrogen blowing into the top so that the liquid doesn't dry out. If the smart cell, the DNA liquid dries out, it'll kill the movies because the enzymes will all die. So we seal it, and we blow in the nitrogen, and we keep that going, and that'll usually last us for our 3-hour movies, on our really long movies that we take. And then 3 hours, so you can just do the math. 2.5 nucleotide attaches times 3 hours is 2.5 per second. That tells you how many we can run. The reagent drawer, so you have about 4.5 degrees C. Some of our runs, if you're running like 16 cells, you might end up running like for 24 or 36 hours, so we have to keep the reagents stable. Other parts of the subsystem, we're just trying to maintain at 25 to prevent condensation and keep that clean. The stage itself, as I mentioned, where the smart cell is anchored, it's critical that we maintain it. Right now, I think we're tagged at about 20 degrees C, but it has to stay there. And the reason is the enzyme will speed up or slow down if you heat it, right, or cool it. And so we have to watch that temperature as well to make sure that we don't get too far off. Because if the enzyme goes too fast, and I'll talk about this in a little bit, you can get your the kinetic signature, which the software is triggering off of, on the incorporations of bases will go off, and then you'll get false base calls or incorrect. It'll just mess up everything. So you have to watch the enzyme to make sure. Really, a lot of what we're trying to do is protect the enzyme forever so that it doesn't die. Because if the enzyme keeps sequencing, we keep getting data. If the enzyme gets too much O2 in the suspension, or the temperature goes off, or a lot of times we're fighting laser power. We have photo damage on our enzymes, which will kill them after some amount of time, so we're trying to always draw down our laser power or come up with more impervious enzymes by putting types of shieldings and stuff with different molecules. All sorts of games we do with that. That's key. And then we have dehumidifiers and conditioners just to make sure that the rest of the system isn't condensing.